Hello everyone, my name is Priti Sarkar. Welcome to my YouTube channel. I'm a recent graduate from University of Rajshahi in biochemistry and molecular biology. And however, today we would like to discuss about a new interesting topic, and the topic is called DNA Microarray. And we will also discuss about how it helps to identify differentially expressed gene between two samples. One sample is control sample or normal sample, and other sample is disease sample. So this process is really crucial to understand the mechanism of disease, and we can also discover new biomarkers through this process. So this process is really important. You might be wondering, what are differentially expressed genes? So differentially expressed genes are the genes that show a significant difference actually between two samples in activity. So if I give you an example, you will be clear. Like just imagine some certain genes that turned on in cancer cells but off in normal or healthy cells. Like we know that some genes are not expressed and they are tightly regulated. So that's why if the genes are turned on, so we can consider them as differentially expressed genes. So these are the differentially expressed genes. So how DNA microarray identifies the differentially expressed genes? So it is a step-by-step process actually. So in the first step, it is called sample collection. So you need to collect the tissues from two different samples. As I said before, that one is the normal sample or healthy sample, and other is the disease sample, and you can consider this as the cancer sample. The second step is called the mRNA extraction or RNA extraction. So in this case, all you need to do is to extract the mRNA from both samples. Here, mRNA represents the genes which are being actively expressed. So in the third step, it is called reverse transcription to cDNA. So why it is called reverse transcription? Because in this process, the mRNA is converted to complementary cDNA. So why it is called reverse transcription? Because we know that in the transcription process, DNA is converted to mRNA. And in this case, the mRNA is converted to complementary DNA. And this process is really crucial because DNA microarray can only detect the DNA, not the RNA. And why, you might be wondering that why cDNA? Because cDNA mirrors the gene expression of the mRNA, but it is more stable and easier to work with. In the fourth step, it is called the labeling of the cDNA. So in this step, you need to label the cDNA with different fluorescent dye. For example, one sample could be labeled with green dye and other sample could be labeled with red dye. So it is easier for you to distinguish the two samples during analysis. Well, after labeling the cDNA, you have to apply those labeled cDNA into a DNA microarray which has thousands of spots, and each spot has DNA probes. And what are DNA probes? So DNA probes are actually the complementary of the known gene sequences. And how it works actually. So if the gene is expressed, then the complementary DNA will be attached to the DNA probes. If not, they cannot be attached. And then wash the DNA array so that the unbounded DNA will be removed. Now a laser is used to scan the microarray to detect the fluorescent dye. So now we can visualize this data or heat map or scatter plot on the computer screen so that we can interpret the result. So in the data interpretation, if the color is green, we can say that the gene is highly expressed in normal tissue, whereas in the disease tissue, it is not much expressed. And if the color is red, we can say that the gene is overexpressed in disease tissue. Whereas in normal tissue, it is not like that. So this is the difference. If the color is yellow, we can say that the gene is equally expressed. And in case of black, we can say that no gene is expressed in normal tissue or disease tissue. So this technique is useful for understanding the mechanism of disease like cancers. And we can know that the genes are overexpressed or underexpressed. By comparing gene expression, we can identify the biomarkers or potential drug targets. So this process is really useful for this drug discovery also. So that's why I think this is the end of our session. I hope you enjoy the video. So don't forget to like and subscribe.